Hello everyone, my name is Dr. Malti, lecturer in Biology, GSI, PU College, Mysuru. In this session, I am going to discuss about biology question paper pattern. This is applicable only for this year, that is 21-22 only. Now, coming to we already studied, there are 5 units, unit number 6, 7, 8, 9 and 10. Unit 6, that is the reproduction. Here we understood reproduction in organism. We understood what is meant by reproduction, what is the definition for reproduction, and what are the 2 different types of reproduction. The one is said to be asexual, second one is the sexual, and then what are the different types of asexual reproduction with examples, and in case of plants it is said to be vegetative propagation, and we learned there are different types of vegetative propagation with example. Coming to the sexual reproduction in organism, here we understood there are 4 stages of sexually reproducing organisms, and then we understood the events of sexual reproduction: pre-fertilization, fertilization and post-fertilization. This is about the first chapter, that is reproduction in organism. So, in this chapter reproduction in organism, 1 mark question is only 1, 2 marks is 1, 3 is 1, there is an absence of 5 mark question. Then coming to the sexual reproduction in flowering plants. Here we understood parts of a typical flower, that means a flower consists of calyx, corolla, androecium and gynoecium. The important part is androecium and gynoecium. We understood in detail androecium. When we take a transverse section of dithecous anther, you can see there is a presence of 4 chambers. What are all the different parts that is present in dithecous anther, and then how there will be a synthesis of pollen grains, and the process is said to be microsporogenesis. Next is gynoecium. Gynoecium, which consists of ovary, style and stigma. So, inside the ovary there is a presence of ovules. So, we understood the anatropous ovule, that means it is an inverted ovule. What are all the different parts that is present in the anatropous ovule, and how there will be a synthesis of megaspores. That process is said to be megasporogenesis. And then after the synthesis of megaspore, the megaspore finally get transformed into embryo sac, that is nothing but female gametophyte. Then we understood the pollination. It is a transfer of pollen grains from anther to the stigma of the gynoecium. There are 2 types of pollination. The one is said to be self-pollination, second one is said to be cross-pollination. As the name itself indicates, self, that includes only 1 plant, cross-pollination that includes 2 plants, autogamy and then allogamy. And then we understood what are all the different types of contrivances, that means adaptations to achieve only self-pollination and cross-pollination. In case of cross-pollination, that is very important, that is outbreeding devices. There are 5 different types of outbreeding devices with example. Then we understood pollen pistil interaction. Coming to the next is the double fertilization. It is present only in case of angiosperm. Here the 2 male gametes participate in 2 fertilization, therefore it is said to be double fertilization. And what are all the changes takes place after fertilization, that is said to be post fertilization. So, here in the sexual reproduction in flowering plants, you will get 1 mark question is 2, 2 mark question is 1, 3 mark question is 1, so 5 mark question is 1. Very important weightage chapter. Then coming to the human reproduction, it is the 3rd chapter. Here we understood male reproductive system, female reproductive system, and the transverse section of mammalian testis and mammalian ovary, and the structure of mammary glands. And then we understood gametogenesis, synthesis of gametes in the reproductive gonads. The one is said to be spermatogenesis, second one is said to be oogenesis. And then we understood the process of fertilization, insemination, before that gametogenesis, insemination, capacitation, acrosomal reaction, amphimixis, and then there will be a formation of zygote. That zygote now enters into the process called, the process is said to be cleavage, and the cleavage is nothing but it is a division. Then there will be a formation of first stage in human being, that is said to be blastocyst. That blastocyst is get attached to the inner wall of the uterus. That process is said to be implantation, and the further development takes place inside the uterus. After the implantation, what happens? There will be a formation of organic connection between mother's blood and the fetus blood, that is said to be placenta. And then what are the functions of placenta? What are the different types of hormones that is related during pregnancy? And after 9 months, what happens? 9 months, few days, there will be a parturition takes place, followed by the lactation period. In human reproduction, there is no 1 mark question. You will get only 2 mark question 1, 3 mark question 1, 5 mark question 1. Then coming to the reproductive health. Here very simple and very small chapter, contraceptive methods, spacing method and then permanent method, and what are all the different types of contraceptive with example is very important. And then infertility, inability to conceive a child is said to be infertility, and then if there is an infertility occurs in both males and females, what happens? We have to adopt some of the technique that is said to be assisted reproductive technique. Here also we understood there are different types of assisted reproductive techniques. And then here 1 mark question you will get only 1. There is no 2 marks and 3 marks, you will get only 5 mark question 1. Then coming to the second unit, genetics and evolution, and the 5th chapter is principles of heredity. So here we understood what is meant by heredity, what is meant by variation, and what is meant by Mendel's law of inheritance, Mendel's first law dominance, and then law of purity of gametes and law of independent assortment with example, that is monohybrid and dihybrid. Along with that we understood the incomplete dominance. Incomplete dominance, appearance of intermediate character in the F1 generation due to the presence of partially expressed dominant gene. And then we understood multiple alleles. What are multiple alleles? Pleiotropy, polygenes with examples. And then we understood chromosomal theory of inheritance, and then very important one is the sex determination. Sex determination, XX, XY type, XX, X0 type, ZZ, ZW type. Very, very important topic. And then we understood Morgan experiment on linkage. What is meant by linkage, what is meant by crossing over, and what is meant by recombinants, how the recombinants are formed, and the Morgan experiment on linkage is very, very important in this chapter. Here we understood gene disorder, any variation in the gene, that is said to be gene disorder. We understood color blindness, hemophilia, color blindness is also known as daltonism, hemophilia, and then cystic fibrosis. And then coming to the next is the chromosomal disorder. The chromosomal disorder means that is nothing but any variation in the chromosome number that results in a particular type of a syndrome, that is said to be chromosomal disorder, Down syndrome, Klinefelter syndrome, and then Turner syndrome. In this chapter, you are going to get 1 mark question is 2 and the 2 mark question is 2, 3 marks is 1 and the 5 marks is 1. Most weightage chapter. Then coming to the next, molecular basis of inheritance. Here we learnt what is meant by DNA, what is meant by RNA, what are all the components that is present in both DNA and RNA, and how these DNA components that are assembled together to form an elongated polynucleotide chain that is nothing but DNA, and we understood a Watson and Crick model, very important, and the semi-conservative method of replication. Why this is said to be semi-conservative? Because the 50% of the DNA strand that is obtained from the parent and the remaining is newly synthesized, and we learned an experiment to prove that the replication was said to be semi-conservative. And then we understood the transformation experiment, very important, Griffith experiment along with an Avery et al. Experiment. How there will be a R strain that is get transformed into S strain that results in pneumonia. Pneumonia is caused mainly due to Streptococcus pneumoniae. And then we understood the transduction experiment, picking up of foreign DNA from the surrounding media, that is said to be transduction. Then coming to the RNA, what are the different types of non-genetic RNA, messenger RNA, ribosomal RNA, and then transfer RNA, and the structure of these 3 very important. Then is the genetic code, group of 3 nitrogen bases that is found on mRNA, that is said to be what do you call genetic code. And then what are the diagnostic characters or salient features of genetic code, that is also very important. And then we learned transcription unit, a portion of the DNA which undergoes transcription, that portion is said to be what do you call transcription unit. In that, promoter, structural gene, and then terminator. And then we understood the central dogma, that is flow of genetic material from the nucleus into the cytoplasm, where there will be a synthesis of protein takes place. So that the central dogma, which contains 2 important processes, transcription and translation, both are very important. Transcription, synthesis of mRNA from the DNA in the nucleus. After the synthesis of mRNA, that mRNA now enters into the cytoplasm, where there will be a synthesis of protein takes place. Transcription in both prokaryotes and eukaryotes is very important. Translation, that is nothing but synthesis of proteins in the cytoplasm. Then is the lac operon concept. It is the regulation of gene action in Escherichia coli, which consists of structural gene, control gene. Control gene that controls the structural gene, so that in some times what happens, the gene becomes passive, so there will be no synthesis of mRNA. In some cases it becomes active. Due to the presence of lactose, the gene get activated. That is said to be regulation of gene action in Escherichia coli. And then we studied Human Genome Project. What are the goals of Human Genome Project? What are the diagnostic characters of Human Genome Project? And what is the methodology of Human Genome Project? And then we understood the last concept, that is nothing but genetic fingerprinting. How there will be identification of an individual, that is mainly based on the repetitive units that is present in human beings. Now, in this chapter, molecular basis, you will get only 1 mark question is only 1, and there is an absence of 2 mark question. So that 3 mark question is 1 and 5 mark question is 2. This is also more weightage chapter. Then coming to the evolution. In this chapter, we understood what is meant by evolution, convergent, divergent evolution, adaptive radiation, Big Bang theory, Miller's experiment. Next is human health and disease. Here we understood what is meant by health, what is meant by disease, and then what is meant by pathogens and what is meant by infectious disease, non-infectious disease, and some of the diseases along with the causative organism. For example, malaria is due to Plasmodium vivax, and then ringworm disease is due to Trichophyton rubrum, and then pneumonia is due to Streptococcus pneumoniae, like that. And then we understood the structure of antibody, that is also very important, and then we learned life cycle of Plasmodium, and then we learned immunity, two different types of immunity, active and passive immunity, what is meant by vaccination, what is meant by allergy, or what is meant by autoimmune disease, what is meant by AIDS. Then we learned cancer, causes for this, and then detection and diagnosis, and finally is the treatment. Next we learned alcohol and drug abuse. Here we learned some of the drugs that is extracted from the plants. For example, opioids that is extracted from Papaver somniferum, and then cannabinoids that is extracted from Cannabis sativum, and then morphine, and then LSD. So these are all some of the drugs, and what are the effects of these drugs, that is also very important. Effects of alcohol and prevention and control. So in this chapter you will get the 1 mark question is 2 mark question is 1, 3 mark is 1, 5 marks is also 1. Next is strategies for the enhancement of food production. Here we learned dairy farming, poultry farming, apiculture, plant breeding, and then different steps that is present in the plant breeding, disease resistant varieties, and then insect resistant varieties, and the plant breeding for the improvement of food quality, that is single cell protein, biofortification is also important. And finally we understood tissue culture. So here in this strategies for enhancement of food production, there is no 1 mark question, only 2 mark question is 1, and the 3 marks is 1, and the last is 5 mark question is 1. Next, microbes in human welfare. Here we understood microbes in household products, industrial uses, and then biofertilizer, some of the antibiotics, sewage treatment, and then microbes in the production of biogas and the BT cotton. Here coming to the questions here, 1 mark question you will get 2. Next is biotechnology principles and processes. Here we understood genetic engineering, tools for genetic engineering, that is vector, host cell, and some of the enzymes, restriction enzymes, both exo and endo, nuclease enzyme, and then DNA ligase and the bioreactors, and the separation of DNA fragments by electrophoresis, that is also very, very important. And the cloning vector, microinjection, and then gene gun. Process of recombinant DNA technology. Here there are different steps. First is the selection of DNA, isolation of DNA, cutting of DNA into fragments, then is an amplification, then it is the introduction into the host cell, then the last process is the downstream process. In this chapter you will get 1 mark question only 1, 2 marks is 1, there is no 3 mark question, and the last is the 5 mark question is only 1. Next is biotechnology and its application. Here we learned some of the applications in agriculture, pest resistant varieties in plants, BT, cotton, and then RNA interference, and then in synthesis of insulin in a large scale, and then gene therapy, ADA, that means adenosine deaminase deficiency, BT cotton, molecular diagnosis, transgenic animals, ethical issues and biopiracy. Here you will get only 1 mark question 1, there is no 2 mark and 3 mark, only 5 mark question is 1. So, 1 mark question is 1 and 5 mark question is 1. Coming to the last 10th unit, that is ecology, in that organism and population. Here we learnt abiotic factor and some of the response to abiotic factor, population growth, in that exponential and logistic growth, and then different types of population interaction. Population interaction in that predation, competition, parasitism and then commensalism. You will get only 3 mark question is 1 and 5 mark question is 1. There is no 1 mark and 2 mark question. Next, coming to the ecosystem, productivity, decomposition, energy flow, food web and food chain, standing crop, ecological succession and nutrient cycle. You will get, there is no 1 mark and 2 mark question, only 3 mark question is 1 and the 5 mark question is 1. Next is biodiversity and conservation. Here we understood biodiversity, species, genetic and then ecological diversity, pattern of biodiversity, loss of biodiversity and conservation of biodiversity, and in situ and ex situ conservation. In this chapter you will get 2 mark question is, 1 mark question is 1 and the 2 mark question is only 1. There is an absence of both 3 and 5 mark question. Last chapter is an environmental issues. Here we understood air pollution, prevention and control of air pollution, electrostatic precipitator and CNG, compressed natural gas. ಇಲ್ಲಿ ನಮ್ಮ ಕಾಲೇಜಿನ ಬಸ್ ಮೊದಲನೇದಾಗಿ ಮೈಸೂರಲ್ಲಿ CNG ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗಿದೆ ಅಂತ ಹೇಳೋದಕ್ಕೆ ನಾವಿಲ್ಲಿ ಹೆಮ್ಮೆ ಪಡ್ತೀವಿ. ವಾಟರ್ ಪೊಲ್ಯೂಷನ್ ಆ್ಯಂಡ್ ಇಟ್ಸ್ ಕಂಟ್ರೋಲ್, ಡೊಮೆಸ್ಟಿಕ್ ವೇಸ್ಟ್, ಇಂಡಸ್ಟ್ರಿಯಲ್ ವೇಸ್ಟ್, BOD, biological oxygen demand, biofortification, biomagnification, eutrophication, agrochemical and its effects, greenhouse effect, global warming, ozone depletion, deforestation and reforestation. In this chapter, there is no 1 mark and 2 mark question, you will get only 3 mark question 1 and then 5 mark question 1. This is about the second PUC biology question paper pattern for the year 22 only because of the pandemic. And here you will get 1 mark question 15, 2 mark question 10 and then 3 mark question 10, 5 mark question 15. So, total marks is 140. You are going to write only for 70 marks. This is about the important aspects present in each chapter along with the blueprint, that is 1 mark, 2 mark, 3 and 5 mark questions. So, the total marks is 140. For this you will be writing for the examination only 70 marks. I wish you all the best for the examination. Do well in the examination. Thank you.